Hello and welcome to this Feedinfo webcast discussing advances in livestock immunity and the role nutrition can play. I am Ollie Theocaris, Head of Perspectives Content at Feedinfo, and I will be your moderator today. Our webinar has been developed with our partners Herbonis today, and we do thank them for their support in developing this piece of content. So I firstly want to explain a bit about how this will work if you've not seen a Feedinfo webinar before. We've pre-recorded this session, but our speakers are online right now to take questions in the Q&A chat box on the right-hand side of the screen. So if you post a question in the questions tab, they will be able to type you a response. So please ask away throughout the webcast and you'll be getting some responses through there. We're also going to be releasing a number of polls throughout, so if you want to share your opinions, but also see what others in the industry are thinking, do participate in these in the poll tab. So I would like to begin by introducing our speakers. So we have three experts with us today who are going to give us the inside story about what is going on in the field of livestock immunity and what we can do to improve it. So we have with us today Dr. Catherine Buehler, Head of R&D at Herbonis Animal Health, Professor Bernd Kaspers, Professor in Animal Physiology at Ludwig Maximilians University Munich, and Dr. Jan Dirk van der Klies, who is the Head of the Technical Department at Herbonis Animal Health. It is great to have you all with us today, and I'm really looking forward to getting a really great overview of this subject and some insight into the topic. So I want you to each introduce yourselves first, our lovely speakers, so our viewers can learn a bit more about you. So Catherine Buehler, can you start by telling us about yourself?
Welcome to everybody out there. My name is Catherine Buehler and I'm working as Head of R&D at Herbonis. I'm a biologist by training and did my PhD in pig nutrition, both at ETH Zurich here in beautiful Switzerland. I started working at Herbonis 5 years ago, and before that I was also working in a feed mill doing feed formulation for all kinds of animals.
Fantastic, thank you. Very well placed to speak on this subject today then. And Professor Bernd Kaspers, over to you. Can you just tell us a little bit about yourself?
Yeah, of course, it's a pleasure. Well, I studied veterinary medicine in Munich and graduated in animal physiology, essentially. Got my PhD in animal physiology, and then I moved on to immunology and spent some time in the United States to research coccidiosis in chickens. And then I returned to Munich and have been working on the immune system of chickens essentially since then.
Fantastic. So we're going to get some really good insight into what is going on within animal immune systems from you. And finally, Jan Dirk van der Klies, can you tell us a little bit about yourself?
I started in Herbonis about 1.5 years ago as a head of the technical department. On the one hand side, I'm developing new products together with the R&D department. On the other hand, transmitting the data from the R&D department towards product positioning and new products to the clients together with the technical department. Before Herbonis, I've been working in the industry already for quite some time, in the feed additive industry. And before I started, I did work at different research institutes in the Netherlands, where I was working generally on poultry nutrition and on consultancy of nutritionists on a global scale. My education has been in Wageningen, where I did a PhD on mineral nutrition in poultry.
Thank you for that overview, and thank you everyone. We have some really great minds who are going to give us their opinions on this subject today. So we are going to discuss livestock immunity today. We all, I believe, know how important livestock immunity is. It's the first line of defense against health issues for animals. And we know that, there are things that can influence an organism's immune system, and one of them is nutrition. So we're going to take a bit of a deep dive today to understand the up-to-date research focused on immunity in animals, what we're learning in that field, and also learn a bit more about the sorts of things nutritionists can do to influence animal health. So I want to kick off with my first questions to understand really the scale of the challenge. With antibiotic-free production and no antibiotics ever production becoming more and more common in livestock animal husbandry, a good resilient immune system has really become a key priority. So Janda van der Klees, I'm going to come to you first. Can you tell us about the factors that can modulate the immune system in livestock?
I would say that since we had the ban on in-feed antibiotic, the prophylactic use of in-feed antibiotics in Europe, we were more or less panicking to see how we could deal with that challenge to the industry. And it's only by then that we became fully aware of the importance of immunocompetence of our farm animals. Antibiotic-free production requires generally more resilient animals that are able to deal with the many pathogens that come from the environment. And as veterinarians, nutritionists, and farmers, we have quite a wide toolbox available to improve the resilience of the animals. Examples are, for instance, the vaccination programs we all are aware of. We are also breeding animals to optimize the productivity under variable conditions like hot or cold environments, clean or dirty environments. And we are studying nutritional adaptations to stimulate intestinal health or vaccination responses. All of these nutritional adaptations include feed additives like, for instance, probiotics or phytogenics, trace minerals, and of course vitamins, where today we focus on vitamin D as an example. Of course, we should not forget animal management and biosecurity, which have become more and more important. Now, prophylactic use of antibiotics is no longer an option, or in case of illnesses, fallback scenarios of its curative use should be minimized.
So there you mentioned vitamin D. So to most of us, vitamin D is well known as the sunshine vitamin that of course affects calcium utilization and is used for treatment in the elderly for osteoporosis. But in recent years, vitamin D has been linked, sometimes controversially, with a variety of other functions such as fertility and autoimmune diseases, more recently for its role in development of proper immunity against COVID-19. So, Catherine Buehler, I want to ask you here, what is the backstory and where did this interest in vitamin D come from?
Well, to answer this question, allow me to go back in time, especially to the year 1848, and the location is a hospital in London. This hospital was used to treat tuberculosis patients, and the doctors there, they gave their patients cod liver oil. Maybe you remember it, that you had to eat this cod liver oil as a kid. From the treated tuberculosis patients, so the patients that received about 3 times a day this cod liver oil, 18% of them did not progress in tuberculosis, but 19% deteriorated and some even died. That sounds like a lot, but compared to the control group, that means the tuberculosis patients that did not receive the treatment, the figures are still very good, because in the control treatment, only 5% of the patients stopped progressing with the tuberculosis, but more than 30% of the tuberculosis patients died. It was not much later in the 1860s that light treatment and also cod liver oil treatment became a standard treatment for tuberculosis. And in 1903, there was even a Nobel Prize for treating tuberculosis with UVB. So you see this link between UVB, and you mentioned the vitamin D as a sunshine vitamin, and tuberculosis is not a new discovery. Later on, with the discovery of the antibiotics, this knowledge moved into the background, and people didn't think about it. And when more of the vitamin D metabolism was discovered, that there's more than just vitamin D, that there are other forms of vitamin D, that there is a vitamin D receptor, that was about 50 years ago. This connection to this to the immune system became interesting again. And especially when it was discovered that the vitamin D receptor is also present on immune cells, then researchers started to think, okay, if the vitamin D receptor is present on immune cells, there must be a connection to immunity. And especially in the last decades, with progression in molecular methods, with progression in analytical methods, and of course also now with the COVID pandemic, this connection between vitamin D and immunity has gained a new momentum. And actually at the moment, most of the research with what we call these non-classical effects is done in regard of vitamin D and immune system.
That's really interesting. I always love hearing about the history of how we got to where we are today. And clearly, it's been a real focus for many of late, given the global circumstances. So let's think about the immune system. I think we've all got an understanding that the immune system is a tool of the body to fight diseases. But of course, we have Professor Caspers with us, who is an expert in immunology. So it would be really fantastic, Professor Caspers, if you can give us a brief overview of the immune system and the ingredients required for a good immune response, so we have that background information.
Yeah, it will be a pleasure, even though it's pretty tough to do it in just a minute or two. But in general, the immune system is set up in such a way that we have a rapid response system, which is called the innate immune system, and then we have a sustained system, which is called the adaptive immune system. What is important is shown here in the first slide, which is that we are challenged, and all animals are also challenged by a vast number of different pathogens, such as viruses, bacteria, fungi, and parasites, endoparasites, helminth, ectoparasites. And because we have such a vast number of different pathogens with different biologies, the immune system has to be pretty complex to combat all these different pathogens properly. So in particular, the adaptive immune system has brought about a number of specialized lymphocytes, and one which is well known nowadays with the coronavirus infection, I think, is a B lymphocyte, which is producing antibodies. And those antibodies can neutralize viruses, can inhibit bacteria, and can also combat parasitic infections. But not all these infections can be combated by B lymphocytes. And in particular, B lymphocytes need help to do that. And they will get that help through T lymphocytes, which are shown here, which are still called CD4 T lymphocytes. Some of you may have heard that before. It's just a marker for those cells. And these cells produce soluble mediators called cytokines. We talk a lot about cytokines, and also in the context of nutrition factors, which may modulate cytokine production and thus the role they play in the immune response. So, for example, as shown here, one type of CD4 cells will produce cytokines called interleukin or IL-4, IL-5, IL-13, which will help B cells to make good antibodies. And on the other hand, these cytokines also will help the body to combat parasitic infections. But that is not enough to combat, for example, viral infection or bacterial infections. And thus, specialized T lymphocytes producing, for example, interferon gamma or interleukin 17 to combat viruses and bacteria have evolved in evolution. And all these different types are now present in our immune system and in the immune system of our domestic animal species. Well, you asked me about what makes a good immune system. And on the next slide, I would like to emphasize what is actually happening, and that is that we have a situation shown here on the left-hand side where we have a healthy, normal gut. But if that gut gets infected, for example, by salmonella or by viruses, immune response will be mounted, and you can see this here, massive infiltration of cells. All these brown dots are cells moving into this tissue. And these cells will start an inflammatory response by, again, secreting soluble mediators of the immune system like interleukin-1, interleukin-6, and TNF-alpha. And this will tip the balance towards inflammation. And a good immune system should be able to mount that response rapidly and to mount it to a degree where it is needed to control the infection. But at the end, this whole system should also tip back to a homeostatic situation, so the balance has to come back. And for this, we again need particular cells which are able to do this. These cells produce cytokines, again, like interleukin-10 and TGF-beta, and one of the cells doing that is the so-called regulatory T cell, Treg. And this leads to an anti-inflammatory response. And if this doesn't work, we get inflammation, overwhelming inflammation, and that's part of the problem in COVID patients. If this is not properly controlled, the virus might already be gone, but the inflammation is still going on and is not regulated, and that is the major problem there. So what we need is a good regulation of the system, and there are numerous factors impacting on that. One of them is vitamin D, which has been shown to play a role in the development and function of such T regulatory cells.
Thank you for that response. But you highlighted there, obviously, the diversity of the immune system, but we know that all organisms face more or less the same environment with similar immunological challenges. So, Professor Caspers, is the immune system comparable among different species, including humans, cows, swine, and poultry? Can you tell us how similar this is within different organisms?
Yeah, I mean, overall, the systems are very similar. They have evolved over a long period of time. And even though, for example, birds and mammals segregated some 300 million years ago, the basic features and the cell types are all present in birds and in all of our domestic animal species. But there are some significant differences, and not everything we learn in, for example, in mice can be translated to humans right away. This is, for example, obvious in vitamin D research, because some results found in mice do not really apply to humans and the other way around. But in general, we have very similar systems working in a very similar manner. One difference is shown in the next slide, which is the transfer, for example, of maternal antibodies from the mother to the offspring. While in primates and maybe in our dogs and cats, due to the particular type of placentation, the antibodies are transferred from the mother directly through the placenta to the offspring. In other species, as we all know, these antibodies are transmitted by colostrum. And so ruminants on horses have to take up colostrum right after birth as early as possible in order to acquire those maternal antibodies through the milk. On later time points, this will not be efficient anymore, and so it's very critical to get that early on. And in birds, obviously, they have no placenta and they don't produce milk, so the way to transfer it is via the egg. Secreted by the hen in the egg yolk, and then the developing embryo will take it from the egg yolk and take it up into its circulation, will be protected this way. And in the next slide, I just want to emphasize that there are also other differences, minor differences, because they lead at the end of the day to the same capability for responding to pathogens. But for example, development of those very important B lymphocytes in mammals takes place in the bone marrow. While in birds, we have a very unique organ called the bursa fabricius, which is shown here on the right-hand side. And the bursa fabricius is really unique to birds. You don't find it in any other villi in the animal kingdom. So even though we have those two different organs, the end result is we get antibodies, and we get good antibodies to protect our animals. And on top of that, for example, in birds, we don't have lymph nodes. this is still a little bit of a mysterium because we do not really know how they mount and where they mount their immune response. That is clearly up for research over the next period of time.
Thank you for that overview. So clearly there are some differences for those in animal husbandry to be considering when developing plans to boost animal immunity and health. Now, Jan van der Klees, I'm going to come to you. We've already touched on vitamin D and its influence on the immune system, but can you give a bit of a summary on the current research in this area within livestock production?
I think it was great, the explanation of Professor Kaspers of the different aspects of the immune response, the immunocompetence of animals. And when we do research, we generally do that using challenge models, where we are only studying specific components of the immune response on the efficacy of nutrient utilization. The improved antibody response is generally a low-cost response, and therefore often preferred as a response of the animal to a challenge. A much more expensive response is dedicated to reduce the inflammatory response after intestinal respiratory infections. Understanding factors like on vitamin D metabolism would give us a possibility to better utilize its potential to stimulate the immunocompetence of livestock under variable conditions. But of course, what I've said, it is very often using challenge conditions and only giving an answer under very specific circumstances.
Thank you for that overview. I think that's a really good picture of kind of where we're up to and where the research needs to go next. But I want to now get some opinions from a few of you on some subjects we haven't yet addressed in this area to get some potentially conflicting thoughts. We'll see. So we've gained some insight into the immune system over the past half hour, but how does what we know of the immune system fit in with modern livestock production? And Jan van der Klees, I'm going to come back to you on this. Can you offer your thoughts?
As we already indicated during this webinar, although being essential, the inflammatory response to combat pathogen invasions is generally an expensive one. And what we are trying to do is to limit the inflammatory response to improve the resilience of the host. And it's more or less like a patient. If you are ill in bed, you try to prevent a high body temperature. You try to prevent fever by taking few aspirins. And everybody knows that when you have taken the aspirins, you already feel better because the fever is coming down. A lower fever also reduces the mobilization of muscle protein, body proteins, during the infection. And of course, when you don't mobilize it, you don't have to rebuild it afterwards. In livestock production, we're more or less doing the same. We are defining resilience to an infection, often as a reduced adverse effect of the infection on production performance, so less mobilization of body protein. And from the nutrition point of view, we are discussing the nutritional cost of immunity, cost that we try to keep as low as possible without putting the immunocompetence at risk.
It's a very good point. And of course, we know the economic side of things is always very important for those involved in animal husbandry. Professor Kaspers, anything to add on this point?
Well, I think this is a very critical point. We need to, in order to maintain productivity of our animals, we need to have resilient animals, and we have to have efficient control of microbial infections. So saying that breeding resilience into our domestic animals will be very important in the future. I think there are some good examples. For example, layers. Layers have been bred to produce high numbers of eggs, more than 300 in a row. And to perform this great challenge, I think these birds have to be very resilient, and the immune system has to be very strong. Otherwise, the birds wouldn't survive such a long period of high performance. But we don't understand these mechanisms really in detail, and that is partially due to the fact that we don't have the tools yet So we have to invest more to better understand how those really resilient birds are able to cope with all these challenges and at the same time to still have a high productivity rate.
I think there's certainly a balance to be found there. And actually that leads quite nicely into my next question, looking to the future. So I want to find out what you each think are the biggest challenges when considering immunity, and what do we have left to learn? I know we've already touched on a few areas, but I'd love to get your thoughts on this. So this time we'll start with Catherine. Can you give your thoughts on this, please?
I can try. In my personal opinion, one of the biggest challenges when talking about immunity is the complexity of the immune system and the fact that it is constantly interacting with the environment. As I mentioned earlier, there are a lot of studies looking at vitamin D and the interaction with the immune system. However, it is unclear what was first. It's sort of an egg or hen question. Was the low vitamin D status the reason for the onset of the disease, or was the onset of the disease the cause for the low vitamin D status? In addition, it seems that the different components of the immune system, as we learned about them by Professor Kaspers, they act at different times differently, and it is unclear whether the different components, for example, when we talk about vitamin D, act on these different components at the same time in the same way, or whether they're only relevant at a certain time point. And this makes also doing trials or repeating trials very, very complex, very difficult, because this time affects the variety of cells involved in the immune system. They make it really hard to pinpoint a certain effect, a certain observed effect to a certain cause.
Thank you for giving your thoughts on that. And I'm going to ask the same question to Professor Kaspers. What do you think are the biggest challenges when considering immunity, and what do we have left to learn?
Yeah, I think one major challenge is the correlation between what you asked previously, the correlation between a good immune response and productivity of the animal. And to measure an immune response, we need a lot of tools to do so. This has to be a large toolbox because we have to deal with so many different components of the immune system. And in particular, in our domestic animal species, we don't have that toolbox yet. Not if we compare what they have in the mouse world or in human medicine, for example, right now. But still there. I mean, if you look at the COVID infection again, everybody's talking about antibodies. Well, very few people, only specialists, I would say, talk about T lymphocytes. And they might also be of critical, or they surely are of critical importance in the control of the infection. And in particular, in the control of the inflammation which is started during the infection. But still, we very rarely measure that, because measuring T-cell responses, for example, is very time-consuming and is restricted to specialized laboratories. So for the future, we have to develop much more tools, and tools which can be applied, in particular in our case, in the animal world, can be applied in the field, or in field studies, which I think would be very important. And I fully agree with Catherine. Timing is very important. Kinetics are of critical importance, and that is always a challenge for any animal trial.
Some really good ideas there. Some other things for us all certainly to be thinking about and maybe investing in as well. And finally, Janda van der Klees, can you give me your thoughts on this subject and what you think we need to start thinking about and researching?
I would say that an aspect that we didn't put on the table so far is that we are extending the production period of livestock as much as possible. If an animal is producing for a longer period of time, the smaller the investment that you do raising an animal to become productive. Evidently, if the animals are getting older doing so. One important example is, for instance, in laying hens. In the past, we grew them to about 75 weeks of age, and right now they are getting 100 weeks or even older. And what we have shown is that the activity of the enzyme systems that are used to activate vitamin D into its bioactive form are getting less when the birds grow older. And this is also what we know from human, that elderly human also have problems with, for instance, bone calcification. And it's interesting to speculate on the consequence of age on the immunocompetence of the older laying hens. This might be one of the causes why many laying hens have a high mortality, many breeds have a high mortality when they are getting towards the end of the production period. And maybe by improving the vitamin D metabolism or anticipating on the vitamin D metabolism, we can reduce that mortality at the end of production.
It's a really good point. We certainly need to be thinking about, animals at each stage of their life and what we can do to support them. And of course, with layers, we are expecting them to survive as long as possible and support us. I've got one final question to wrap up this discussion. I want to know your piece of advice, your take-home message for our audience. And let's go back to Jan van der Klees on this one. Would you be able to start us off?
We all know, or we have learned, that the immunocompetence is a delicate balance between the different aspects of the immune response. And we have shown, or we have illustrated, that vitamin D metabolites play a central role in immunomodulation in livestock. However, we do not yet understand how an optimum immunocompetence works. What's an optimum immunocompetence, if even present for a total population? And for the time being, one of the targets in animal nutrition is to limit the overshoot of the innate immunity to improve the resilience to different infections.
Very good. Good take-home message there. And Professor Caspers, over to you next. What's your take-home message for our audience?
Yeah, I fully agree. I mean, it's a complex task, as I alluded to before, and we need good experimental models in order to study the different effects of nutrients on the immune system. We need challenge models in order to do so, because we might not see much under homeostatic or normal conditions, but we need to challenge the animals, which is adding another layer of complexity to such studies. And I think we need to develop more tools in order to investigate this and to help people in the field of nutrition, in the field of animal breeding, and in the field of vaccine development to help them to apply those tools to address their questions and to better understand how the immune system works, how it can be improved, and how it can be supported, in particular, for example, again, with vitamin D. I mean, that is well known that it's critical to have a proper level of vitamin D to have a healthy and functional immune system, as introduced in the beginning with the example of tuberculosis.
Yeah, you raised some really good points there too. I think what is particularly interesting there is the fact that we have many groups who are relying on this information. As you mentioned, we could pass findings from these experiments on to so many different people involved in livestock value chains and could really benefit a number of groups in our attempt to boost immunity across many different groups. So I'm going to come finally to Catherine Bühler. Can you finally give us your take-home message?
I think the scientific community today agrees that there's evidence that vitamin D has an influence on the immune system. However, it surely is not the silver bullet that solves all the problems. It is, however, and also in my opinion, an important part that can be used to modulate and to fine-tune the immune system so that we have a really balanced outcome. And it has been mentioned before, we need to have a quick response, but it cannot be overshooting. And in my opinion, vitamin D has the potential to figure out this balance, to support this balance in the immune system.
Some great advice there too. Thank you for your insight to all of you. And I think it's a really nice point to end on. So it's clear from what we've heard today, this is a complex area and we are still learning more. And there's a lot more that we need to invest in and research. But given the circumstances and the changing legislation globally, the challenge of boosting immunity in livestock farming is just not going away. And it's something we should all be prioritising. So it's been really fantastic to get all of your thoughts on this subject. Our speakers are still online, and they will continue to be so for another 10 minutes or so. So if you do have any follow-up questions, do keep putting your questions to them. But I would like to take this opportunity to thank our speakers. You've all brought a really great knowledge and insight to today's session. Thank you to everyone who's tuned in and contributed to our polls and asked questions. We hope you found it interesting and that you tune in to future sessions from Feedinfo.